Hi everyone, welcome back to my channel. In this video, I'm going to talk about the concept spermatogenesis. The word spermatogenesis is derived from the Greek word in which sperma means seed or sperm and genesis means production. So as the name indicates, spermatogenesis is a process of production of a sperm. Spermatogenesis takes place in the male gonad called as testis. So testis mainly consists of a highly convoluted tubule called as seminiferous tubule. Inside this each seminiferous tubule, spermatogenesis takes place. So this seminiferous tubule is lined by the epithelial lining called as germinal epithelium. Germinal epithelium made up of germ cells. So this germinal epithelium gives rise to sperm by the process of spermatogenesis. In some of the animal like mammals and mollusks, these germinal cells are interrupted by some of the somatic cell called as Sertoli cell. Sertoli cells are the long columnar cell which is present in between the germ cells. These Sertoli cell will anchor the differentiating cell and it will nourish the developing sperm. So along with the germ cell, these Sertoli cells are also helps in spermatogenesis. Spermatogenesis is a continuous process and for our convenience it is mainly divided into two steps. First one is formation of spermatid and second one is spermiogenesis. Formation of spermatid is also divided into three phases, that is multiplication phase, growth phase and maturation phase. We will discuss it one by one. The first step in the spermatogenesis is formation of spermatids. The male germinal cell which produces a sperm is called as a primordial cell. Primordial cell. It is also called as a primary germinal cell. This primordial cell will be having large nucleus and this primordial cell will undergo all these phases to produce spermatids. The first step undergone by the primordial cell is multiplication phase. In the multiplication phase, this primordial cell undergo continuous mitosis to produce a cell called as spermatogonia. This spermatogonia will be having 2n number of chromosomes. And there are mainly two types of spermatogonia, that is type A spermatogonia and type B spermatogonia. These type A spermatogonia are the stem cells which undergo continuous mitotic division to produce again spermatogonia. On the other hand, type B spermatogonia enlarge to produce a primary spermatocyte. Ultimately, it produces spermatozoa. The spermatogonia which is produced as a result of multiplication phase, now it undergo next phase called as growth phase. In the growth phase, this spermatogonia cell accumulates large amount of nutrition which results in enormous increase in the size of the cell. And now it is called as primary spermatocyte. And this primary spermatocyte have 2n number of chromosomes. This primary spermatocyte now undergo next stage called as maturation phase. In the maturation phase, the primary spermatocyte undergo first meiosis or meiosis first. There are many processes takes place in the meiosis first. Like there will be pairing of the homologous chromosome or synapsis takes place. The homologous chromosomes split longitudinally. There is a formation of chiasma by which the homologous chromosomes exchange their genetic material or crossing over takes place, etc. So after all this process, these primary spermatocytes divide to produce two secondary spermatocytes. Since the meiosis is a reductional division, these secondary spermatocytes will be having n number of chromosomes. Now this secondary spermatid undergo second meiotic division to produce two spermatid each. Since the second meiotic division is an equational division, the number of chromosome which is present in the spermatid remain same, that is it have n number of chromosome. This spermatid cannot act as a mature sperm. So spermatid should undergo one more step called as spermiogenesis to form a spermatozoa or sperm. And the last step is spermiogenesis. Spermiogenesis is a process in which the spermatid will differentiate to produce spermatozoa or sperm. The sperm or spermatozoa is highly active or mobile cell. To acquire this character, the spermatid will discard most of its content and it undergoes certain changes during spermiogenesis. The main changes in the spermiogenesis includes changes in the nucleus, acrosome formation and changes in the centriole. We'll discuss it one by one. The main changes occur during the spermiogenesis is changes in the nucleus. The nucleus of the spermatid has large amount of water. So during spermiogenesis, there will be loss of water from the nuclear sap and that nucleus will shrink to attain the new shape. Anyhow, the shape of this nucleus will differ from one species to another species. But this shape is the one which determines the shape of the sperm head. Apart from that, in the nucleus and the nucleolus, the RNA content will be greatly reduced and the chromatin content of the nucleus will be concentrated. So these are the main changes that takes place in the nucleus. The next very important changes during the spermiogenesis is acrosome formation. Acrosome is a cap-like structure which is present near the anterior region of the sperm head. This acrosome consists of a proteolytic enzyme. This proteolytic enzyme of the acrosome helps in the penetration into the egg. And this formation of acrosome takes place during the spermiogenesis. This acrosome is formed from the Golgi apparatus. We'll see how the acrosome formation takes place. At the beginning, the Golgi apparatus accumulate the anterior region of the sperm nucleus. In the next step, few vacuoles of this Golgi apparatus fuse to form a large vacuole and it occupies a central region. So soon after the formation of large vacuole, a granule will develop inside this vacuole and it is called as a proacrosomal granule. Later, this proacrosomal granule will enlarge and it will attach to the sperm nucleus to form a acrosome. And this membrane of the acrosomal vesicle forms a double membrane cap-like structure in the anterior region of the sperm head. That is how exactly acrosome formation takes place. One more major changes that occur during the spermiogenesis is changes in the centriole. The two centriole which is present in the spermatid, it will occupy the position just below the sperm nucleus. And these centrioles are present one below the other. The anterior centriole is called as proximal centriole and the posterior centriole is called as distal centriole. This distal centriole changes to form a basal body and this basal body gives rise to axial filament or flagellum of the sperm. The mitochondria of the centriole will fuse and it is spirally coiled around this axial filament. The mitochondria and the basal portion of the axial filament is present in the mid-piece of the sperm. These are the major changes that takes place during the spermiogenesis that transforms spermatid into spermatozoa. That is all about the steps involved in the spermatogenesis. I hope you like this video. If you like this video, please hit the like button and subscribe to my channel. Thank you.